Hello and welcome back to my YouTube channel. In this video, we'll go through two topics. One is the osmosis, another is the active transport in much detail. Firstly, we'll discuss osmosis. Later, the focus of this video will be shifted to exploring active transport. So let's jump into the lecture. So you must ask what osmosis actually is. It is the movement of water molecule from their higher concentration to their lower concentration across semi-permeable membrane or selectively permeable membrane. We have got another definition as well, which tells it is the movement of water molecule from low solute concentration to the high solute concentration across selectively permeable membrane. So how it occurs? Let's learn it through the diagram. We have got diagram. It has got two chambers, chamber A and chamber B, which are separated by the selectively permeable membrane. Chamber A is filled with sodium chloride solution, whereas chamber B is filled with pure water. We know that because of the sodium chloride ions or sodium chloride solute in the chamber A, the concentration of water becomes lower than the chamber B. So we know that osmosis is the movement of water molecule from higher concentration to the lower concentration across semi-permeable membrane. So let's connect this diagram to the second definition as well. We learned that osmosis is the movement of water molecule from low solute concentration, because the chamber B is filled with pure water. It doesn't have solutes in it. So the movement of water molecule in this case will be from chamber B to the chamber A, from low solute concentration to the high solute concentration across semi-permeable membrane. So it must be clear to you, I hope. The movement of water will continue. Okay, so the movement of water will be from the chamber B to chamber A till the point where pressure across this membrane will develop, which now causes the stoppage or cessation in the osmosis. Okay, because the movement of water causes the rise in water level in this chamber A. So due to the rise in water, the pressure will be developed. Enough pressure will be developed which causes the stoppage or cessation in osmosis. Okay, and this pressure is known as osmotic pressure. What is osmotic pressure? Pressure at which osmosis stops. There will be no osmosis in this case at this pressure. So osmotic pressure is a colligative property, which means that it does not depend upon the size or mass of particle. It depends only upon the number of particles in a given solution. Okay, so osmotic pressure is a colligative property. So we express number of solutes or the concentration of solutes in a solution in terms of osmolarity and osmolality. Before understanding these two concepts, we must go through osmol, what osmol actually is. We have got another concept known as osmol. Okay, osmol is a measurement which expresses the number of solutes. Okay, it expresses the number of solutes in a solution. Okay, in a solution which contributes to its osmotic pressure. Okay, so this was the definition of osmol. Let's learn it through the diagram. We have got a container. Okay, this container is filled with sodium chloride solution. So we have filled 1 mole of sodium chloride solution. This 1 mole of sodium chloride will now be dissociated into their respective ions. Okay, sodium ions will be there, chloride ions will be there. These both ions will be contributing to the solution's osmotic pressure. Okay, so now 1 mole of sodium chloride solution is equal to 2 osmol. Okay, because this sodium chloride in a solution will be dissociated into their respective ions. That's why 1 mole of sodium chloride is equal to the 2 osmoles. Another container, we have got another container which is filled with the glucose solution. Okay, solution of glucose. The 1 mole of glucose is equal to 1 osmole. Why is it so? Because glucose is not an ionic compound or ionic substance, so it does not dissociate into its ions. So that's why 1 mole of glucose is equal to the 1 osmole. Okay, so I hope this is clear to you. So now let's move on to the osmolarity and osmolality. Let's start with the osmolarity first. Osmolarity is the osmole of a solute in a liter of solution. Okay, whereas osmolality is the osmole of solutes in a kilogram of a solution. Okay, so this osmolarity and osmolarity is are essential for the maintenance of body's fluid balance. Okay, it is the solute concentration in a solution in a body's fluid which determines the movement of water and solutes across the different compartments. If the osmolarity of blood decreases, which causes the movement of water molecule from the blood into the cells, causing the swelling of cell. On the other hand, if osmolarity of blood increases, which causes the movement of water outside the cells, therefore causing the shrinkage of cells. Okay, so the osmolarity of blood should be maintained, so there must be some mechanisms by which osmolarity of blood is balanced. It is balanced if the osmolarity of blood increases, which stimulates the thirst. Okay, so it stimulates thirst mechanism, thirst stimulus as well. There will be the thirst stimuli, okay, which causes the which indicates there is the need of more fluid in the body. There will be the also release of ADH from the pituitary gland as well, which also indicates the increased water need or fluid need in the body. Okay, so this ADH in the renal tubules causes the reabsorption of water. Okay, so now therefore causes the balance in blood osmolarity. Okay, so these regulatory mechanisms, we will study them in the renal physiology. We have just discussed them in a very brief manner. So now we are done with the osmosis. Now let's talk about active transport. We studied in our previous video that active transport is the movement of molecules or ions from their low concentration to their higher concentration with the input of energy. Okay, so if the source of energy is directly by the cleavage of ATP, then this process is known as primary active transport. If energy comes through the indirect source, then this process is known as secondary active transport. Let's learn primary active transport first. Okay, the best example of primary active transport is the sodium potassium ATPases or sodium potassium pump, which is operated almost in all cells of the body. Okay, so this pump has got two units. One is the alpha unit, another is the beta unit. The function of beta unit is not that much significant, but it anchors the protein complex to lipid bilayer. Okay, so it only performs the function of anchoring in this case, whereas alpha unit has got two binding sites for the potassium at its outer portion, whereas it has got three binding sites for the sodium at its inner portion. Okay, it has got a molecular seizure as well, which perform the function of ATPases. As soon as the binding sites bind with their respective ions, this ATPases gets activated. You must remember, as soon as the binding sites bind with their respective ions, ATPase is activated, which causes the cleavage of ATP, into the ADP plus inorganic phosphate, and an amount of energy is released. This amount of energy is used for the flipping of this transporter due to the flipping of this transporter, 3 sodiums are thrown out and 2 potassiums are brought in. Okay, so because 3 sodiums are thrown out, there will be the net loss of 1 charge, due to which the inner side of membrane becomes negative, more negative, whereas the outer side of membrane becomes more positive. You must remember. So because it is involved in the generation of potential difference across this membrane, so this transporter is electrogenic in nature as well. So it performs the function of electrogenic. Okay, so we know that wherever sodium goes, it takes water alongside as well, and the membrane is less permeable to the sodium as well. So whenever the volume of cell increases, these sodium potassium ATPases or sodium potassium pump is activated or implanted within the cell. Okay, and now throws 3 sodiums outside the cell. Because 3 sodiums are thrown outside, this sodiums also takes water alongside as well, so therefore causes the balance of cell volume, also maintain the cell or cellular volume. Okay, so these are the 2 functions of sodium potassium ATPases. One is the maintenance of cell volume, whereas other is the electrogenic nature. Okay, electrogenic because 3 sodiums are thrown out, 2 potassiums are brought in. There is the loss of 1 net charge, due to which inner side of membrane become more negative, whereas outer side becomes more positive. So that's why it is the electrogenic in nature. Let's move on to the another example of primary active transport, which is the calcium pump. Okay, so there are the 2 sites where these calcium pump are implanted. One is the cell membrane, another is the membrane of endoplasmic or sarcoplasmic reticulum. You must remember, this calcium pump is implanted within the cell membrane as well as the membrane of endoplasmic or sarcoplasmic reticulum. We know that the concentration of calcium in the extracellular fluid is 10,000 times more than the intracellular fluid. So how this increased concentration in extracellular fluid is achieved? This is achieved by these pumps. In the first pump, the calcium is thrown outside the cell, whereas in the second pump, in the pump which is implanted within the membrane of endoplasmic reticulum, the calcium is brought into the endoplasmic reticulum. So that's why, which causes the increased level of calcium in the extracellular fluid, whereas decreased level in the intracellular fluid. Okay, so this was all about primary active transport, where the source of energy is directly by the cleavage of ATP. Okay, there is another type of active transport as well, which is known as the secondary active transport, in which the source of energy is not direct by the cleavage of ATP. It comes through the indirect source. How? We know that the sodium potassium ATPases was throwing 3 sodiums outside, and we also know that sodium is the membrane's permeability for sodium is also much lesser. So an electrochemical gradient of sodium is generated, which has got increased sodium concentration outside the cell, whereas decreased sodium concentration inside the cell. So this sodium always tends to come inside the cell, but cannot pass through this lipid bilayer. There must be some transporters implanted within the cell, which causes the movement of sodium from outside to inside. So this sodium, if moves with another molecule, okay, so this sodium adds the movement of another molecule. If it adds the movement of molecule in the same direction, then it is known as symport, and if adds the movement of molecule in opposite direction, then it is known as antipode. Let's start our discussion with the symport first. Symport, also known as co-transport. The best example of symport is sodium glucose co-transport. Okay, we take carbohydrate diet. If a person takes carbohydrate diet, the carbohydrate is digested within the small intestine into the small units. If it is the starch, then it is digested into the glucose. This glucose must be taken up by the cells, by the intestinal cells. These intestinal cells has got sodium glucose co-transporters implanted within their membrane. Okay, so we know that the sodium is moving from higher concentration to the lower concentration. Whenever any ion move move along the concentration gradient, there will be the release of energy. So this energy is used to move less glucose, which is present in the lumen of the intestine, and low glucose and higher glucose level in the cell. Okay, so now glucose is moving against the concentration gradient and sodium is moving along the concentration gradient. So this energy which is released by the movement of sodium along the concentration gradient is now used to drag the glucose from the lumen of intestine into the cell. These sodium glucose co-transporters are also implanted within the PCT cell as well. They also causes the reabsorption of glucose in the lumen, in the kidney as well, the lumen of PCT. Okay, so this must be clear to you. Another example of import is sodium amino acid transport as well. Okay, which also operates in the same manner as the sodium glucose co-transporter operate. So there is the same mechanism behind it as well. So this sodium amino acid co-transporters are implanted within the cells of small intestines, within the microvilli of small intestine, also within the PCT cells as well. So this it must be clear to you. We have got another type of secondary active transport as well, which is known as counter transport or the antipode. The best example of antipode is the sodium proton antipode. Okay, in which we know that sodium is in the higher concentration in the extracellular fluid, whereas it is in the lower concentration in the intracellular fluid, whereas protons are higher in the extracellular fluid, where they are lower in the intracellular fluid. Okay, so they are low in the intracellular fluid and higher in the extracellular fluid. So in this case, sodium is moving along the concentration gradient. So the energy which is released in this case is now being used to drag this proton outside the cell. So this is the best example of counter transport or antipode. We have got another example as well, which is the sodium calcium counter transport as well, in which sodium is moving inside the cell, whereas calcium is thrown outside the cell by the energy which is released by the movement of the sodium ions along the concentration gradient. So with this, we are done with the active transport as well. I hope you enjoy this lecture. Thank you for watching.